Hello everyone, this is Rahul. Welcome back. In first part of our discussion on apoptosis, I presented an account of what is apoptosis, what are the causes of apoptosis, differences between apoptosis and necrosis, what are caspases, how caspases are activated, how caspases are repressed, and what are the important signaling pathways of apoptosis in a vertebrate system. We also discussed death receptor pathway of apoptosis in great detail. In present discussion, that is part of apoptosis, we are going to see mitochondrial pathway of apoptosis in great detail, how apoptotic cells are cleared, apoptotic pathway in nematodes, and also I will provide some practice questions and references for more reading on apoptosis. So let us begin our discussion. Now let us learn about the mitochondrial pathway of apoptosis. Mitochondrial pathway is also known as intrinsic pathway. Mitochondrial outer membrane permeabilization, or MOMP, is a key feature of this pathway. Upon MOMP, various soluble proteins in the mitochondrial intermembrane space diffuse into the cytosol. Among these proteins, holocytochrome C plays a fundamental role in activating caspases. As shown in this slide, MOMP is tightly regulated by the BCL2 family proteins, which are central to the mitochondrial pathway of apoptosis. BCL2 family proteins share up to 4 BCL2 homology domains, or BH domains. They also share structural features with some bacterial pore-forming proteins, including the diphtheria toxin B chain. There are 4 BH domains, that is BH1, BH2, BH3 and BH4. There are 3 general subfamilies of BCL2 proteins that induce, directly cause or inhibit MOMP. These include, first, the anti-apoptotic BCL2 proteins. These proteins possess BH4 domain along with other domains. Its examples are BCL2, BCLXL, as shown in red in present slide. The second category of BCL2 family proteins are the pro-apoptotic multi-domain proteins. These carry BH1, 2 and 3 domain but lack BH4 domain. Its examples are BAX and BAC. The third category is of the pro-apoptotic BH3 only proteins. These carry only BH3 domain. Its examples are BID, BAD and BIM. You can see the complete mitochondrial pathway in the write-up on the right side of this slide. Here you can clearly see that BAX and BAC, 2 of the multi-domain proteins in the BCL2 family, are responsible for MOMP and probably form the pores by their oligomerization on the mitochondrial outer membrane, through which the proteins of the mitochondrial intermembrane space diffuse out. BAX and BAC in general do not function to cause MOMP unless activated. The BH3 only proteins, mainly BID and BIM, activate BAX and BAC oligomerization. The anti-apoptotic BCL2 proteins, including BCL2 and BCLXL, bind to the activator proteins, for example BIM, and prevent their function, and also bind to active forms of BAX and BAC. In this way, the anti-apoptotic BCL2 family members prevent MOMP and prevent apoptosis. Other members of the BH3 only protein subfamily, for example BAD, block the functions of anti-apoptotic BCL2 family members, that is inhibition of the inhibitors, hence promoting activation of activator BH3 only proteins like BIM, which in turn activates BAC and BAX to cause MOMP. Hence these are de-repressor or sensitizer BH3 only proteins. As a result of MOMP, soluble proteins including holocytochrome C in the mitochondrial intermembrane space diffuse into the cytosol diagram, where they interact with cytosolic proteins. In the cytosol, holocytochrome C binds to APAF1 protein, that is apoptotic protease activating factor 1, inducing it to bind with a molecule of DATP so that it may oligomerize to form a complex of 7 APAF1 molecules as shown in this diagram. It forms a cup-like structure. Here it is shown as a cup. This structure is called an apoptosome, the center of which has exposed card domains. I have already mentioned about card domains in previous slides, which bind to the card domain of the initiator caspase 9. The binding of caspase 9 molecules to the card domains of the apoptosome forces the caspases to dimerize, thus activating them by induced proximity. I have also explained what is induced. Before moving further, let me clear one thing to the students who are viewing this slide, that cytochrome C contains two parts. One is protein part or peptide part, and the another part is heme, or it contains an iron moiety in between the heme structure. The protein part without its heme part is called apocytochrome, and the entire protein molecule along with its cofactor is known as holocytochrome C. In fact, it is prosthetic group. Heme is the prosthetic group in this complete peptide molecule. Once activated, caspase 9 cleaves and activates the executioner caspases 3 and 7 and apoptosis. Apart from holocytochrome C, some other mitochondrial intermembrane space proteins are also released in cytosol during MOMP. Two of these proteins are SMAC, also called Diblo, and OMI, also called HTRA2. These antagonize the caspase inhibitory activity of IAPs. I have also explained in previous slides what are IAPs. Its example is XIAP. In both OMI and SMAC, the extreme N-terminal sequence called the AVPI motif is responsible for inhibiting XIAP. This was the mitochondrial pathway of apoptosis. It is a little complicated. You can go to the previous slide and continue listening whatever I have explained, and you can note down to avoid any kind of confusion. So also it will help you in revising the Also, there is link between the death receptor pathway and mitochondrial pathway of apoptosis. Caspase 8, activated upon ligation of death receptors, cleaves the BH3 only protein BID, resulting in its activation. BID then triggers BAX and BAC to cause MOMP, thereby engaging the mitochondrial pathway of apoptosis. Therefore, BID acts as a link between the two apoptotic pathways. Apoptosis was discovered in nematodes. It was first of all discovered in Cynorhabditis elegans, in which 131 out of 1090 adult somatic cells undergo apoptosis during development. This is very precise and accurate process, and cells die predictably at a defined time and place in each worm, and this developmental cell death proceeds through apoptosis only. Apoptosis in nematode follows a simple pathway with similarities to the mitochondrial pathway of apoptosis in the vertebrates. The complete pathway you can understand from the handout I have given in this slide on the right side. Here, the most important thing is to remember the homologs. For example, the anti-apoptotic BCL2 homologue of vertebrates in C. elegans is SED9. Similarly, APF1 homologue is SED4. Procaspase 9 homologue is SED3. So these things you should remember because questions are generally asked regarding these homologues in CSIR examinations. The clearance or burial of the dying cell is the ultimate function of apoptosis, as the cell is removed with almost no indication that it had ever been there. This occurs by phagocytosis or cell eating, either by professional phagocytes like macrophages or dendritic cells, or even cells, or even some other cells like epithelial cells. The importance of efficient clearance of apoptotic cells is underscored by the characteristics of apoptotic cell death that probably contribute to this process as the cell is packaged for removal. DNA is chopped into small pieces for facilitated digestion, cells fragment into bite-sized morsels, and the cell generates signals to solicit its engulfment. The latter process can be summarized as eat me signals and the disruption of don't eat me signals. So what are eat me signals? Eat me signal involves externalization of phospholipid called phosphatidylserine to the outer leaflet of the cell membrane. In normal condition, phosphatidylserine is present in the internal leaflet or inner leaflet of the cell membrane. The externalization of phosphatidylserine occurs when enzymes like ATP-dependent flipases and scramblase are activated during the process of apoptosis. After externalization of phosphatidylserine molecules, some other extracellular factors like MFG-E8 and annexin-1 are recruited over the externalized phosphatidylserine molecules. The recruitment of these extracellular factors lead to tethering of integrin molecules present on the surface of phagocytes. This tethering allows phagocyte to come closer to the dying cells, which facilitates their clearance by engulfment by the phagocytes. Sometimes in normal cells, phosphatidylserine is externalized without any apoptotic signal. So these cells are not engulfed by phagocytes because of the presence of a don't eat me signal on their surface in the form of a molecule called CD31 antigen. MFG-E8, annexin-1, these are used as markers of apoptosis in experimental studies. So we see that clearance of apoptotic cells is a highly regulated process. Now let us move towards further discussion where we are going to see what happens if apoptotic control mechanism. Apoptosis is a highly regulated process, but sometimes these regulations fail. So let us see what happens when failure of apoptotic controls occur. Failure of clearance of apoptotic cells is observed in an autoimmune disorder known as systemic lupus erythematosus. The characteristic feature of this disease is the formation of butterfly-like patches on the face of the patient, as shown in the right photograph in this slide. In another case, inappropriate activation of apoptosis occurs. In many neurodegenerative diseases and ischemic injury, this inappropriate activation of apoptosis is seen. Blockage of apoptosis occurs in different types of cancers, which leads to the survival of cancer cells. Now at the end of our discussion, let me provide you few questions through which you can assess yourself regarding the concept of apoptosis and your learning output. The first question is, in apoptosis, the cell destroys itself from within and avoids leakage of the cell contents into the extracellular space. What might be the consequences if programmed cell death were not achieved in so neat and orderly a fashion? The second question is, one important role of FAS and FAS ligand is to mediate the elimination of tumor cells by killer lymphocytes. In a study of 35 primary lung and colon tumors, half the tumors were found to have amplified and overexpressed a gene for a secreted protein that binds to FAS ligand. How do you suppose that overexpression of this protein might contribute to the survival of these tumor cells? Explain your reasoning. You can write the answers of these questions and email me at my email ID, that is livelycolors@gmail.com. livelycolors@gmail.com. To gain more insight into the process of apoptosis, you can follow some of the books. The list of few of the important books is provided in this slide. Thank you.